It is my absolute pleasure to welcome our first presenter. Associate Professor Edwin Hawkins is an immunologist and cancer biologist. He conducted his PhD at WEHI, which is the Walter and Eliza Hill Institute, before relocating to Peter Mac as a National Health and Medical Research Council Peter Doherty Fellow. In 2012, Edwin was awarded the European Haematology Young Investigator Fellowship to train in intravital microscopy at Imperial College in London. Edwin was recruited back to WEHI in October 2015 to bring his international experience to the Institute and establish a research program studying hematopoiesis, inflammation, and cancer, which he continues to drive today. Thank you and welcome, Edwin.
Thanks so much for that introduction, Laura, and apologies for all the long, really annoying words in there. You did a really good job with them. But I think what I want to touch on is I'm going to show you some research today, which has been a passion of mine for a really long time. But as Laura outlined, I did always have in mind doing this project that I'm going to share with you today, even back when I was a lot younger. And this is the reason why I went to London. And what you're looking at in front of you is actually a picture of bone with big clusters of myeloma forming plasma cells in there. I'm going to tell you about this story today and how it relates to human disease. And I just want to highlight that the work that I'm showing today is from Joel Rhymes and Joy Liu, who are PhD students in the lab, Raymond Yip, who's a postdoc, and Jeremy Yu, who's a consultant hematologist at Peter Mac, who's now performing his PhD with me. So what we do in my laboratory is we find different ways of imaging cells in live tissue. We quite often do this using mouse models or preclinical models, and we find different ways of looking at tissue in action. And if you just look at the top left-hand picture that's moving around, that's an image I'm going to talk about a lot today. That's bone marrow. And why I want you to look at that right now is everything that you're looking at in that picture is happening in real time. So there's a big blood vessel, which is that black bit in the middle. The green and the red cells are cells that we've modified to make blood, the hematopoietic stem cells, and everything there that you're looking at is happening in real time as it would in your body as well. So with this great technology, we can look at things that have always been really, really hard to understand, from culturing cells in a dish, and try to get a really good idea of how they function inside the body. So microenvironments is a really, really fancy terminology, and it's a concept that's not really well understood. So let me talk you through it. The idea is that in the bone marrow, where a lot of your hematopoietic cells live and cells that drive myeloma, these plasma cells, that's where they live, we don't know how they work. We know that we get a range of different fates. Some stem cells will make blood, some will make immune cells. And the idea has been that there's little pockets and little areas, microenvironments, that have a unique composition of factors which drive this process. This has been a really interesting concept in blood cancers that have this intimate relationship with bone marrow, because the idea is maybe they move to these areas and they hijack them and take them over for their own good. The reason we want to understand it is we want to understand how certain therapies are failing in these patients, maybe because they form a microenvironment that stops an immunotherapy or a CAR T cell, but also just in general how it works to create an environment that these plasma cells that drive myeloma survive for a really long time. So the concepts at the bottom there, or how do they behave in the bone marrow? How do they hijack these microenvironments, and how do they modify them in their favor? So plasma cells are the thing that drives disease in myeloma, but also lupus, and they're formed from these immune responses that occur usually in your spleen and your lymph node. And then they migrate through a process that we don't understand to the bone marrow, where the fascinating thing about them is during an immune response, they're maintained there for your entire lifetime. So these plasma cells are sort of already evolved to be really long-lived. They'll survive for the lifetime of the individual in the bone marrow. Now, we know that they'll circulate through the blood and get there. And then when they hit these spots, that's a plasma cell drawn in the middle of bone marrow, there's osteoblasts and osteoclasts, and they make bone. And we know that the disruption of how they function is a really important thing in myeloma. We know that they disrupt the function of those cells, and that's why a lot of patients get fractures. We also know that they interact with the blood vessels. But there's a range of immune cells as well that have been proposed to be the things that help to support these plasma cells that survive for a long time. So of course the concept is we can figure out the cell type to delete and get rid of. Maybe then we can target these plasma cells better. So the question is there a niche or a microenvironment? Is it intrinsic versus extrinsic? And what that means is it genetic mutations that really drives this and how they function in plasma cells? Or is it this information from the cells around them? And then do they grow better? Do they survive better? Or is it a little bit of both together that combines over a really long time to cause the expansion of these cells? So here is a picture of what myeloma looks like in the bone marrow of an animal. This is a mouse model where we can mimic what happens in the mutations that drive this disease. We can transplant them into mice. And the left-hand picture is one of these TAL scans of the bone marrow of a live animal when we've transplanted in myeloma. And the really interesting thing about myeloma is it forms these distinct foci, these focal areas, where essentially the plasma cells will infiltrate, grow, and force everything else around them. But the right-hand side is a videotape, and people like to make these great videotapes of cells moving around in the body, and we do that a lot. And usually they're quite striking when you can see cells moving around. But the plasma cells are very different. They sit there, they form this foci, and they really do nothing. They just wobble around a little bit. And why that's interesting is on the left-hand side here, we've got some red cells that are acute lymphoblastic leukemias and acute myeloid leukemias, either with chemotherapy, without chemotherapy, with different types of drugs that change their interactions. And what I want you to look at on the far left-hand side is how fast these non-plasma cell blood cancers interact with the blood vessels and move throughout the bone marrow. So the fact that plasma cells do that is really, really unique. What we did is we made a really, really smart animal model where what we did is when one of the plasma cells, instead of being a healthy immune protecting plasma cell, becomes a myeloma cell, we targeted them so they would inherit a fluorescent protein. And what that means is then we can look at one cell that's a mutant and all of its progeny and understand what they're doing. So this picture shows it beautifully. You can see these individual clusters, huge clusters of plasma cells. What we usually see in somebody that's been immunized is somewhere between 3 and 5 plasma cells, and these are thousands. And what you'll see is a single color occupies a specific area. So what that tells you is this idea that a plasma cell can move to the bone marrow and make a microenvironment has a lot of strength from looking at it this way. So we know that that's absolutely true. And we've been able to repeat it a lot. The other thing that we know with these mice is they replicate a lot of things that we see in human disease. And that is, it's a problem that we have with disease in older people. Mean age for diagnosis of myeloma is in your 60s. And these mice start to come down with myeloma symptoms about 550 days. That's quite old for a mouse. These mice will live for 2.5 years in the facilities. So these are really, really old mice, and that's what it looks like. But what we now know is that when we take young mice with this model of myeloma and we look in their bone marrow, despite the fact these are 80 to 100-day-old mice, so none of them will develop myeloma for another year, year and a half, they already have these plasma cell clusters in their bone marrow, and we never see these in other models of disease. So what it actually tells us is that these plasma cells move to the bone marrow in a mutated form really early in life, and they're maintained in these little microenvironments or niches there for an extremely long period of time. So it raises the question of when these cells that cause disease actually develop and move to your bone marrow. We now know from studies out of Iceland that when they profiled the entire population, well, about half the population, but they didn't select for whether people had disease or not. They just looked at their serum, and about 5% of people over 50 had an antibody profile like they had MGUS. So what it means is that we think that this shows that a lot of the understanding about when and how myeloma develops is actually not quite right. We think it happens very, very early on. But what it also tells us is that something isn't right about how we're thinking about these cells. So when they move into the bone marrow, the idea has been that they proliferate and grow really, really quickly. But I'm showing you data that says in really young mice, this clone will go there and it won't be a problem for another year and a half. So the nice thing that we were able to do is we're actually able to videotape this over a week in mice with myeloma, and that's shown at the left-hand side. Because we've got really dedicated graduate students, we then could randomize all the information, all the pictures, and get them to count them. And then we could do that over a week because we can anesthetize mice and re-image the bone marrow and look at disease develop. And what we can see is that actually the turnover of these plasma cells over a week, so this is what my students like to call a scientist week. It's Monday, Wednesday, Friday, so they don't have to come in the weekend, despite the fact they often do. But what we could see is that the rate that they grow is actually very, very slow. So on the bottom left, here we have the cell counts of the same population of clones over time, and it can show their turnover is really, really not that fast. One thing I want to seed in your heads today, and I'm not going to talk about it today, is everything that we found from myeloma so far, we also find for another plasma cell disease. It's called lupus, which is an autoimmune disease. Every single result that we find is the same between the two. So on the right-hand side, it's got plasma cell clusters and autoimmunity, then retained over time, and the same results. They're in microenvironments, they're clustered as a single clone, and their turnover is really, really slow. So what we did is we figured out a way that we would then image these cells inside a live animal, and that's shown at the top left, when they developed myeloma or lupus. We would then make a 3D map, which is in the bottom left, of where all those clones were. And then what we could actually do is we could excise the tissue. Now, in mice, we use these landmarks to look at the marrow and identify it really well. But that also means it's very easy for us to cut out and isolate the cells from the area that we looked at and perform genetic analysis. So now what we're doing is we're looking at the RNA that they express, which is the message, and we can look at what's mutated and different in these cells. And what we found was something really, really interesting that I'm going to talk to you about, which is firstly, we hoped to get an idea of certain factors they made or proteins they express that would keep them in these microenvironments. And we actually couldn't find that. It was really, really hard to find. We found a bunch of control genes that we expected to see, and I've highlighted them here. On the left-hand side, that's the data for myeloma. We see things like XBP1, which is a gene that's upregulated when these plasma cells are trying to make lots of antibody. It's a stress-related gene. We see overexpression of this MYC gene, which is commonly mutated in myelomas. And we see plasma cell genes like cyclin D2. When we compare these to normal plasma cells, we find about 74 genes are messed up, differentially expressed, is the way of saying it. When we look in lupus plasma cells, it's about 64. But the fascinating thing here, and the main thing I want to raise out of this, is that of these 64 genes that are differentially expressed in lupus, a completely unrelated disease to myeloma, 62 of them are shared between the two diseases. So what it tells us is that this interaction with bone marrow is probably evolutionary conserved. It's a function of plasma cells in general, which is really good because it probably means if we can figure out what's going on, we can probably broadly apply it to plasma cells in general. So we're very, very excited about that. The other thing is we found a bunch of genes which are called Ly6 genes. They're not very well understood. They code for a protein that is related to immune cells. They've reported to have many, many different functions, but that was really a red herring. And I'll show why it's interesting in a second. Now, one of the things that we're doing at the same time, and this will become apparent why it links all together soon, is we want to understand why these microenvironments form and how they talk to the normal immune system. So this videotape here is clusters of myeloma cells in green and CD8 T cells, which are your main immune cell, in red. And the question we're trying to ask is, how do these myelomas shut down T cell responses? And also, if we're to perform these experiments in the context of CAR T cells interacting with the BCMA antigen, which is the big one, ide-cel and cilta-cel now, or with bispecific antibodies. And what they are is an antibody that targets an antigen on the myelomas, and then they target an activating receptor, CD3, on T cells and activates them. We know the initial responses of patients are really, really good, but then after that, they tend to fail. And this is all work done by Jeremy Ur, who's the consultant hematologist that I raised before. So what we could do, if the video plays, is we can ask this question about, well, what do myeloma cells do to exclude T cells? If they get in there, what do they do to shut down these responses? And how can we understand this better? And the idea is, when we know the genes that are miscoded, or when we know the function of this, we can change how we engineer CAR T or inform the clinicians to do it. but also we can get an idea of any other therapies that we can use to target this. So the first finding is really, really simple, and that is, let me just go back to that, that when we have these T cells in the context of now the myelomas, what you'll see in the bottom graph there, the bottom picture, is the dotted lines are where the myelomas are, and the red are all the T cells. And what we see is when the myeloma gets really, really infiltrated, it pushes all the T cells out. So that's not that unexpected. And the idea based on that has been these areas are immunosuppressive. They shut down the immune system because they probably exclude T cells from these areas. So what we can do now is we've partnered with Bristol Myers Squibb to get an antibody from them that is the same as the bispecific antibody that patients receive in the clinic. We can image the mice on the far left-hand side before they start therapy. On day 3, we can image when they're in the middle of this therapy and look at how the T cells function. And then we can look after day 7 at the relapsing cells. So it really does mimic quite well what we're seeing in patients. And the data that we saw is fascinating. So hopefully this video is going to start in a second. We can now look at the infiltration of these T cells and ask what's going on. So the first thing that people have said is, well, T cells won't be able to get into these areas very efficiently. And when they're in those areas with myeloma cells, they will get turned off in how they function. They won't be able to grow because the myeloma is so immunosuppressive. So we can videotape this in process. So Jeremy's actually interacting with the data. He's circling a few areas. On the right-hand side, we can see massive T cell infiltration in an area where the myeloma is pretty much being destroyed. And on the left-hand side, we can see really nice sort of area where the T cells are trying to interact with the tumor. And what Jeremy's going to do in a second is zoom in on a T cell in action in these areas. And the first thing that we can see is you can actually see that T cell is proliferating. It's rounding up, dividing into 2 and expanding. So the idea that these areas are immunosuppressive and shut down T cell activation and proliferation is completely incorrect. And that's important because we can reject any hypothesis or any work that we want to go into looking at around that. I think Jeremy's got a nice little picture of that in action on the side. You can see that T cell rolls up beautifully. And actually, it's very easy to see dividing T cells and immune cells in living tissue because they always have to stop. They can't migrate. They always have to become a big ball before they divide and then they race off. Okay, so I've told you then there's no problem. The T cells get in, they activate really well, they proliferate in these areas, there's no problem with it. So what is actually going on? And the finding here was quite striking and quite interesting. So day 3, day 4 of the response, when the T cells have started killing, we started videotaping them a lot, and what we noticed is this. So Jeremy's going to zoom up on a T cell in red here. It's surrounded by myeloma. He plays the video, and actually what was quite striking is you see that T cell basically explode and break up into these little fragments that it leaves behind. We've never seen that in any control or anything else that we've ever done before. So this is a unique property of these T cells in these myeloma clusters. So what we actually see, and what Jeremy's doing now is highlighting, actually you can see these red little puncta accumulate throughout the myeloma areas. And what that tells you is that we think the mechanism here is that the myeloma is able to secrete a product that shuts down the T cell responses by inducing death. We don't know if that's activation-induced cell death, anergy, exhaustion, but this really changed our thinking about the theory about how it might work. So of course we wanted to understand that better, but it's a really, really tricky thing to understand. So I've summarized it this way. Scientific data is a big thing of Legos. Cell biologists will like to tell you the different types you have and what they mean. People that do bioinformatics can look at all the gene expression and they can stack up all this data and quantify it. And when we do imaging like I do, we try to take the small amount of Lego bricks, the number of colors that we have to make it work, and kind of show you a picture of how it works. The problem is that we can only look at about 2 to 7 colors at the same time, whereas we've got 27,000 genes to look at. So we've always kind of had this approach of building these houses and understanding it, but we do a terrible job of it. So the way that we're addressing it is this. It's called spatial transcriptomics. And what we can do now is we can take tissue, we can image it and understand where clones are, we can understand where the immune cells are, anything we want to do, and then profile the gene expression in place. Rather than stripping cells out and looking at what genes they express, like I showed you before for our sequencing method, we can look at it in place. So here we have a bone from one of these mice that have myeloma. We can see the different patches of the different clones. And then we can look at all the genes that are expressed in these places. And actually, what we started to see is this, and this is a fascinating finding. So let's just look at this left-hand graph. The idea was that every myeloma cell would remodel the bone marrow in the same way. So there'd be a consistent bunch of cells that they needed to work with to make these microenvironments, probably driven by growth factors that they make and other things. But we never found that. So what we find on the left-hand side is when you look at this Ly6K gene that I was talking about before, it's actually clonally inherited by different plasma cells. So it tells you right off the bat that they're very, very different in the genes they express. But underneath them, we're looking at the profiling of genes that are expressed in the bone marrow tissue itself, not the plasma cells. Cadherin 5, which is expressed on blood vessels, PLVAP, which is also expressed on blood vessels, but other markers for fibroblasts and things. And what we find is that every single one of these areas that have plasma cells is extremely different in the genes that they express. So that was not what we expected, but we're trying to resolve that with other data that we're getting from humans now to understand the question better. The problem with that data is it doesn't have the resolution that we want to have. So I'm really happy to share with you some new data that we've got, and we've applied this all in human. I'm going to share this data with you now of this new technique, which is called Xenium. And we use this to profile the genes in place. So that was an H&E section of bone marrow. I'm clicking on genes to mark bone and blood vessels, which you can see now from this overlay of the morphology, and it works really, really well. And what I'm going to do is highlight now how much resolution we have to understand the genes that are differentially expressed throughout the marrow. So I'm building through, and hematologists love this because it gives a really good idea of all the distribution of the cell types we've always wanted to know. So blood cells, immune cells, stromal cells, here's megakaryocytes that make blood. They look absolutely gorgeous, neutrophils as well. So we have this technology working really, really well. We've just applied it on a bunch of samples that we've got from patients as well. And I think that's what I want to talk about now. So we can profile the genes expressed. We can take samples from patients at different stages of myeloma, smoldering myeloma, MGUS, newly diagnosed, relapsed refractory, and start to look at what the commonalities of the genes are expressed are. So here we have a trophine from our amazing patients that are involved in these studies that donate tissue. And what we can do is profile all the different genes that are differently expressed in the plasma cells, but also the stroma. And we can also do it for mouse, but let's not talk about mouse anymore. So this work was all driven by Jeremy Raymond Yip, who did all the spatial work, and then Andy Chen, who did the analysis. We set up a way of now taking huge cohorts of patients. We've got about 300 trophines. We've only assayed 21 of them for now. Make sure we've got great trophines for these patients. We can then go and look at the genes that are differentially expressed and understand what's going on. And here is a set of data that we've got. As I said, we've got 21 of them now. But all this purple and yellow coloring that you can see here, these are the fluorescent probes to detect all of these genes. So we've got it working really, really well. And this is what the data looks like. And this absolutely blows my mind for someone that's worked on bone for an immense amount of time. This is a control sample from a patient who has follicular lymphoma with no bone marrow involvement. And what we're doing now is dumping every single gene transcript into this sample. 5,000 genes we're looking at, and then we can build up this great encyclopedia of where every cell is, what they're doing, what they're interacting with, and what it looks like. So I'm just going to show you this for a few samples. As I said, we've got larger cohorts of patients now, but you can see the teal are the plasma cells. So at the top are control samples, MGUS, smoldering myeloma, and that little teal population there, that is the plasma cells. And then when we look at newly diagnosed patients, you can see this massive expansion of plasma cells, as we'd expect. We can then look at genes that are differentially expressed between the two. And the outline of the red line there shows this massive expansion of these plasma cells from early stage of disease to late stage of disease. But then we can map them spatially onto these trophines, which is what's on the bottom with the diagnosis standing, CD138. We can look at where all the plasma cells are. And I like this one, MM3 here, because it shows a nice little purple blue area where we actually have the red blood cells that have survived because they're not too infiltrated, and these two distal ends with a lot of stuff going on. And the big finding that we've had is that everything that we see in mouse, we see in human. And I'm going to show it here talking to this trophine that we're looking at. We can look at the relationship of the plasma cells and where they're on the trophine. We can find all the different cell types And then we can perform a gene expression analysis to look at what different clones, what might be doing, where they might be located, and then what's on around them. And what you can see is this red and blue clone that we can find at these different areas that it's refined. And the genes that they express are so different, and their microenvironments are different. And why we think that's important is we think some of those areas are the ones that are causing CAR T cells and people having bispecific antibody therapy. We think that they're completely turning off those T cell responses. And we're about to do a big trial looking at 50 patients before and after CAR T, 40 patients before and after bispecific antibody to understand this. So it's a really, really exciting time. I'm going to finish there. So we know that these plasma cells have this really interesting interaction with the bone marrow. We know it's completely unique. We know that every single plasma cell clone probably has a different microenvironment and not a conserved microenvironment. And a lot of people were disappointed when we talked about this because they said they kind of lost hope and understanding if we could target a certain cell type that was part of that. But upon reflection, I think it really does model what's going on disease a lot closer, and it's a problem that's a lot easier to understand. I think certain plasma cell clones definitely make different factors. They model the microenvironments around them. Some of those are permissive to immune cells, some aren't. And I think actually understanding that might be key for understanding immunotherapy and where we go to next. And as I said, we're about to do a bunch of new spatial work with cohorts of patients, common oncogenic drivers to understand how certain gene mutations drive these microenvironments. And I think it's going to be really, really interesting in the context of these therapies. So I just want to finish by thanking all the people involved. I do get on the microscope a little bit, but nowhere near as much as the people in my lab. But just highlight the work that you've seen today was done by Julie Yu and Joel for all the original imaging studies. Jeremy has done all the bispecific antibody studies in mice and all the human work as well. And all the spatial work is done by Raymond Yip, who I collaborate with, Rory Bowden and Kelly Rogers. And finally, we have great collaborations with Peter Mack. Simon Harrison has co-supervised multiple students in my lab. And that's been really key for us to make sure our results are relevant for patients with myeloma. So thanks very much, Laura. I'm happy to take questions.
Thank you so much, Edwin. The visualizing all of those amazing clonal changes is just, often with myeloma, we often say, it's hard to understand what's going on because there's nothing to visualize necessarily unless you've got bone disease or something. So it's wonderful to see that.
Thanks. And I think, Laura, I'd like to also add that now we've got it to the point where we can transplant myeloma cells from patients into a special type of mouse, and then actually add an antibody in and image their plasma cells in bone marrow. And I think that's the next step, is understanding certain mutations and how they drive that behavior. So the links there to what's going on in human are very, very close.
It's so exciting. I'm imagining a future of, everyone being gene profiled and us looking at your amazing work and having that to really individualize the treatment choices that we actually, choose for these patients. Quick question, and this is probably a little bit simple, but we do know that there are some clonal changes already in myeloma that do drive the decision to treat with one treatment versus another, or might help us risk stratify a patient. Can you elaborate a little bit on what we already know and how that's actually creating treatment decisions in the Australian community at the moment?
Well, I think if I can be bold and take the next extension of that. So, Laura, yeah, absolutely, we know the association of certain oncogenic drivers and what they do. And Laura's 100% right. That changes how they're treated. I think what we want to do now, and I alluded to this before, is perform our work with really good representative numbers from each of those mutations, and then try and understand if they have a common outcome in those microenvironments for every single one of them. So, for instance, you might understand that certain oncogenic drivers drive completely immune permissive niches or immunosuppressive niches. So those patients might need completely different therapy in the context of immunotherapy. Or the other one might be that those common oncogenic drivers give such a diverse range of microenvironments, it's not something that we have to worry about at all. So that's what we're thinking about. We're trying to get funding now to do those sorts of studies. Luckily, because Jeremy is so dedicated to this work, we have this amazing resource of these biobanks of trophines that are donated by patients. They're prepared in a way that we can do spatial on them. So I think we're not far away from asking those questions.
So that's what I would say in the context of that question.
Oh, it's so exciting looking ahead and moving past the standard of care and more towards hopefully a bit more tailored choices depending on what we actually know about each patient's disease. Amazing. Thank you so much for your time.